So hello, everyone. I'm Chris Benko. I'm Connexa's CEO, and I'm pleased to welcome you to today's webinar on the use of actigraphy in clinical research. As many of you joining this webinar know, actigraphy refers to technology based on accelerometers that can be used in clinical research to track a wide variety of different measures, very often without patients needing to be in the clinic. And we're pleased to share with you today some of our experience and best practices on this topic. It's my pleasure to introduce today's speakers. First, we'll have Rob Ellis, who is Connexa's head of data science. Rob has a background in geophysics and numerical modeling, although for the last 6 years as a co-founder of Connexa, he's been working to build the company and its infrastructure and research and development to support clinical studies across a range of conditions with a wide variety of technologies. Followed by Rob, we will have Matt Cantor. Matt heads Connexa's customer-facing operations, so his team is deeply involved in all of the support of the clinical trials we engage with, both from study startup, but also ongoing issues as patients interact with the technology that's required to submit their biometric data to our platform remotely. And we're very pleased to be joined by Abigail Davis. Abigail is a clinical research coordinator at Suburban Research Associates. She's an epidemiologist with MPH from Temple University by training, and we're incredibly happy to have her bring the frontline perspective of interacting with patients across a range of clinical trials, including those that use actigraphy. So thank you all for joining, and I'll turn it over to Rob as our first presenter. Rob, you are still muted.
My apologies. So firstly, a quick overview of actigraphy. I would expect many of the audience are broadly familiar with the topic, but we're going to lay out actigraphy in a certain way to support the rest of the conversation. Actigraphy is a non-invasive way to collect high-resolution accelerometry data, typically from a watch-style device that can be worn on the wrist or worn on the waist or sometimes on the ankle or other places too. The acceleration data that's collected is 3 axes, and that raw data is typically collected at between 30 and 40 samples a second, or hertz, for a fixed period of days. Where the number of days may vary according to the study objectives, the nature of the patient population that you're working with, and so on. However, data collection is really only part of the problem. The second area where I spend most of my time is in processing that data to extract the measures of interest which are required for the particular study in mind. So at the bottom there, you have a picture that lays out the core building blocks of an actigraphy pipeline, from collection of raw data through to the next level, which is to take all of that very dense raw data and turn it into something called epoch data. In many measures, the epochs are like 1 minute in length. You can look at shorter or longer depending upon the algorithms or method of processing that you intend to use. But once you have activity bucketed into epochs, you can then start to process that data. The traditional methods of processing actigraphy data fall into a few buckets. The first is to calculate wake-sleep. Is the patient awake or asleep, depending upon the activity count that's recorded? And then really the processing bifurcates. If your interest is in activity, you will choose one route of processing, and if your interest is sleep, you will choose another. But since the late 80s, early 90s, this has been a burgeoning area of research, and there are multiple choices of algorithms that can be applied for different age groups and tuned and validated against different diseases. Navigating that journey and helping clients with the appropriate study design is something we spend quite a lot of time doing. Next slide, please. So why are the clients that we work with interested in actigraphy? First and foremost, They collect their data in the patient context, often called activities of daily living. This provides an opportunity to capture data as the patient goes about their daily life in ways in which point-in-time assessments or visits to a clinic cannot achieve. With that comes more data. With that comes a need to understand the characteristics of that patient's daily life. But it offers the opportunity for a rich data set to really truly understand whether a drug is changing a patient's ability to go about their daily life compared to before. The second and probably the most important reason people choose actigraphy is its objective. It's capturing the actual motion of the patient. That's very distinct from many traditional EPRO-based measures which measure pain or fatigue or other statements around a patient's perception of their quality of life or mobility and so on. Often you actually need both the objective and the subjective measure, but we'll come to that later. It's also a technology that's vastly matured in recent years, where devices now are small, portable, can be taken home, and with only a small amount of training, a patient can readily provide highly compliant data. So what that really enables in sum is to provide new opportunities to gather both well-established measures such as those for sleep that are long understood in the world of polysomnography to measures around activity which enable a clinical researcher, a study designer, and a pharma sponsor to understand the impact of their drug on the day-to-day life of a patient. Next slide, please. So let's delve more into these different use cases. We lay out 3 core ones here, and then the last one, direct measures, that we'll come to at the end. First and foremost, you deploy actigraphy to determine efficacy. Typical primary endpoints for sleep deployed in conjunction with the diary are now quite commonly used. Activity is a little bit more complicated to dissect. In traditional activity measures, activity is classified according to activity level, sedentary, moderate, vigorous, and so on. That classification turns out to be quite population and age specific. And so it's less mature in terms of its ability to consistently measure an outcome independent of the particular circumstances of the study. Actigraphy can also be deployed as an extension to a clinic assessment. I'm sure many people on the call will be aware that there are a number of mobility type assessments commonly in use today, from the 6-minute walk test that's been around for decades, used in COPD and study of heart failure, to the timed 25-foot walk test, which is a variation, through to specific gait and balance assessments, which are part of a wider assessment, such as Part 3 in the UPDRS. What you're then doing with actigraphy in this use case is you're using the day-to-day actigraphy data collected from the patient to relate it to the evidence from the gold standard test that you've conducted, like the 6-minute walk test and so on. More and more we see people using actigraphy as a corollary measure. So what do we mean by this? If a study intends to measure a patient's fatigue level, it's important to be able to understand if that fatigue level is reflected in their day-to-day activity. It's a reasonable hypothesis, and one we see borne out in practice in deployment. But it's not necessarily as simple as that, as we still don't quite understand if activity level changes as a leading concurrent or a trailing indicator, something we start to think about quite a lot. Lastly, direct measures. So in all of the prior examples, actigraphy data processing and calculation of measures is based upon the concept of the epoch that I mentioned before, and measuring the aggregate activity over a period of time. A lot of the work that we do in Connexa today is focused on the creation of direct empirical measures of gait, such as walking speed, distance, duration of sustained walking, and so on, which is distinct and separate from characterization of activity level. What we'd like to be able to get to is a position where these measures can be used interchangeably off the same device. Next slide, please. So at Connexa, we're device agnostic. We support all of the devices and routinely deploy all of the devices on this slide. When we work with clients, some of the first questions we get are, which device should I choose? Matt will cover more about that later in terms of the operational aspects, but in terms of study design and the measures of interest, the decision points center around the ability of the device to capture data for the duration of the time required and to appropriately provide that data back to our platform. A second avenue of focus is the measures provided. In some cases, a simple daily change in step count or daily change in measure compared to baseline is sufficient. And for that use case, you might not need to deploy the high-grade clinical devices in the first 3 columns of this table. And a Garmin device or something similar might be entirely sufficient. We also have to think about the patient's ability to synchronize and provide us the data. In some disease populations, this can be quite a challenge. Simply put, it's not much use to find out a month from now that a month ago you didn't get the data that you needed. And we work on that kind of problem quite a lot, and Matt will talk more about it. Next slide, please. So on this slide, you can think of it as a dictionary of measures, split by measurement category, for want of a better explanation. On the left-hand side, you have the traditional measures of actigraphy for activity and sleep. And on the right-hand side, you have the gait measures where we focus much of our current research. Both of these measures can be derived from the same device. Now, a device which supports raw transfer of raw accelerometry data. Next slide. So here's where it gets complicated, and a lot of our early work with clients is around taking them through the decisions that need to be made around the points on this slide. So if you're doing a study with an Actigraph device, many actigraphy device vendors will ask you to complete a device and study configuration document. And some of the points on this slide are covered in that document. And what we have to do here is think about what are the study objectives, what is the study population, and so on, and then figure out the best choice of device configuration, algorithms, and so on that fit that population, that are valid, and then we'll provide the measures that are required. So let's take sleep measures, for example, picking on that for now. There are several well-known sleep algorithms out there, Cole-Kripke, Tudor Lock, and so on, all of which can calculate sleep measures. However, those algorithms are validated against certain specific populations. And within that validation comes a choice of settings for minimum levels of activity or minimum period of time that would be expected to be counted as continuous sleep. In some populations, those settings don't work. Same is true for activity measures. An older person's definition of vigorous activity might be very different than a young person's definition of vigorous activity. So what we often recommend when deploying actigraphy for the first time in a new patient population is to do a small validation study to help us and the client understand the settings and configuration of algorithms which is appropriate. That also feeds into operational learnings, which I will let Matt talk more about. Next slide, please. And Matt.
Thanks, Rob. So yeah, the next few slides will be covering how to successfully deploy actigraphy technology into a clinical research study. And at the highest level, the feedback and my commentary would be, take the deployment of these technologies seriously and plan accordingly. So you should be following best practices like the deployment of any technology like a ePro or eCOA data collection technology in that clinical research study. So following a careful design and planning process as you transition once the protocol is finalized into study startup and setup, getting into execution, and then at the end of the completion of the study for data transfers and analysis. You really want to take all these steps into consideration from the start to ensure that the study will be successful in the collection of actigraphy technology. Next slide, please. So for study design, often what happens during this phase is a lot of question and answer. So there's a this in the best case is usually done in collaboration with a sponsor as the protocol is being developed and designed. So, and depending on the stage of the discussion, you could be still evaluating device choice, endpoint or measure selection, and then determining how to best fit a device to a visit schedule or a schedule of assessment. So it's really, really good at this point to kind of keep all items on the table and keep an open mind on how to best deploy these technologies in your study. What measures or what endpoints will help us achieve our goals in the study? What difference do we expect to see in that specific measure, which I'll get into some examples later. Will this be an exploratory measurement, a secondary endpoint, or are we starting to plan as part of a journey towards ideally getting towards a primary measure with the use of these technologies in the future? So depending on the phase of the study, it's really important at this point during the planning to think through the study in front of us as well as future studies and how we can use this technology for different hierarchy of endpoints in the future. Some of the most important considerations around device selection and endpoint and measurement selection really is around the schedule of assessments or the visit schedule. So you want to balance robust data collection with not overburdening the patient as well as the site with wearing the device for too long or too many processes. So what we often see is effective is about a 7 to 14 day data collection period, and typically at a variety of different monitoring periods throughout the study. So a screening and baseline period, and then follow, depending on how long the study is and the condition or disease, maybe additional 1 to 2 week monitoring periods at anywhere between 2 or to 5 periods throughout the rest of the study. This allows you to collect really strong data that you can analyze while not having to the patient wear the device kind of endlessly, because at times you may see reduced compliance if you don't have very distinct wear periods. Maybe next slide, please. So once the device is selected, the measurements are selected, and the protocol is complete, you can move into study startup and study setup. So there are some key activities here that will set the execution and the maintenance phase of the study to be successful. So really detailed guides and training, both for the patients as well as the site teams are really critical. So technology can be new for both of these parties, and you want to make sure that it's as user-friendly and straightforward for them to ensure that they feel comfortable with using a new technology, as well as they're prepared to use it successfully. Of course, with these devices, putting together the appropriate IRB and ethics committee packets with any application screenshots, with any device screens and imagery is critical as you prepare to submit your documents to these different committees. And Rob did mention this, and we'll touch on it in a use case in a little bit, but if time allows, doing maybe a patient pilot is really helpful, especially in populations where this technology hasn't been used extensively previously, maybe in a rare disease. It's good to get a sense of data variability in this certain population, as well as their willingness to use the device, to wear the device. In some cases, an actigraphy endpoint could be used as a screening criteria, so the ability for a patient to wear the device. So going into those studies, you really want to know what compliance to expect from these patient populations, because in some conditions, patients are more tolerable and willing to wear the devices than others. So it's, if time allows, a pilot and the extra work can provide some really valuable insights. Next slide, please. So once everything's been submitted to the ethics committee, and we have technology go live and first patient in, another success or best practice is really putting together very welcoming and informative patient kits. So having everything organized nicely. When they open that kit, it can have the guide, all devices and technologies, really making it welcoming for them, and landing with a strong first impression is really critical, especially with the patient, but also that first site experience when they're opening up what they receive and they see everything nicely organized is critical at this point as well. And then during the maintenance phase of the study, when you're collecting this data, you really want to make it super straightforward for the site teams to be able to view compliance and have appropriate compliance monitoring, so that they are able to partner with their patients appropriately to see if data is being collected as you would expect. So email alerts, detailed dashboards, all of these things are really helpful for the sites as they prepare to collect this data and to support their patients. And I think we'll hear a little bit more on that from Abigail in just a few minutes. Next slide, please. And of course, at the end of a study, you'll be pursuing data transfers, file transfer specs as part of that data transfer process, and then data analysis as well. And I think one critical component to reinforce here is, with data transfers and with actigraphy data in general, it's important to have these conversations upfront. So depending on if the sponsor wants raw data, which could be terabytes upon terabytes of data, or derived metrics, it's really important to understand what the expectation is, where and how that data will be transferred, because large data files don't necessarily always fit into standard systems, and planning should be done accordingly. I think the one other item that at times can cause a little bit of back and forth is around taking a continuous data file like actigraphy data and converting it in an SDTM. Of course, it's possible, but oftentimes there's very kind of defined formats of SDTM files and what the sponsor is expecting. And taking a continuous file just does take a little bit of extra effort to to convert the file to that format, and you should be ready and planned from the start. When the study is being designed and during startup activities, it's really good to start any type of data transfer activities at that time as well. Next slide. So now that we've discussed operationally how to deploy this technology, one of the items that we often discuss is what effect should we expect to see by using an actigraphy technology. And the concept is referred to as the minimal important difference, sometimes the minimal clinical important difference. And more or less what that means is, what is the meaningful change that should be considered either important or beneficial by either a physician or a patient or a caregiver, that would lead the physician or the patient to pursue a different type of care. And this is different and in different populations, different with different endpoints, of course. So for today's purposes, we'll review two examples that may be helpful. The first one in COPD is an interesting example where a step count was collected before and after pulmonary rehabilitation to see. It was a 3-month rehabilitation program to see the change in step count that would be observed there. And in this case, the minimal important difference was calculated with two different approaches, and then triangulated with a variety of different statistical analyses. But after pulmonary rehab, the MID is calculated as between 600 and 1100 steps per day. So if we look at the small end of things, these researchers then looked, okay, if patients do improve by at least 600 steps a day, is that actually clinically meaningful? And what they found is, for the patients that were able to cross that threshold of 600 steps a day following that pulmonary rehabilitation, they followed these patients for 2 years after. They actually had a reduced risk of hospitalization. So you can see there very clear that for patients that are able to walk or able to improve their step count by an average 600 steps a day, had a reduced risk of hospitalization, which is great. Another example in multiple sclerosis is a retrospective analysis. So it was a secondary analysis across 15 different studies, I think about 800 patients, when they were comparing, and this was more of an observational analysis, when they're comparing step count, daily step count versus other clinical measures and patient-reported measures. And in this case, I found a few examples really interesting. So for the multiple sclerosis walking scale, this is a 10-question or 12-question item that then leads to a score out of 100%. And what they noticed is, as patients, as the self-reported data would reduce by 10 points, that would lead to a reduction of about 642 steps a day, which is a great example to have. And then for the PDSS, which is the patient-determined disease scale, it's in this case specific to multiple sclerosis. It's a 0 to 8 scale, where 0 is no movement, dysfunction, and 8 would be bedridden, and then in between you have gait disturbances, cane usage, things like that. What they saw is, on a one point increase on that self-reported scale led to a reduction of about just under 1,000 steps per day, 915 steps per day. So by combining these two analyses, they saw for self-reported scales, the MID had about the MID was about just under 800 steps per day, which is about 14% of average steps for this patient population. And it's really, really helpful when you're planning to use actigraphy in clinical research studies. Obviously, or as you may expect, walk or step count from day to day for patient to patient is pretty variable, but knowing or having an expected difference or an expected clinical important difference or meaningful difference is really helpful when you're starting to think of sample size and things of that sort for these types of endpoints. Next slide, please. This is just another case in a condition known as GLUT1 deficiency syndrome. In this case, we partnered with a sponsor who was hosting a patient advocacy event or participating in a patient advocacy event for GLUT1 deficiency patients. It's a rare metabolic condition where glucose doesn't pass the blood-brain barrier, which leads to a number of developmental delays, seizures in some patients, and movement symptoms and disorders as well. So what the sponsor was hoping to do is evaluate, can we use an actigraphy device to get a sense of functional status of these patients. So through this patient advocacy event, there was a variety of on-site measurements, 6-minute walk test, 12-minute walk test, patient-reported data. But then at the end, the patients were sent home with an actigraphy device, and they wore it for a week. And then there was also patient matched controls, healthy matched controls, excuse me, that were recruited to participate in this as well. And what you see on the right side is a 24-hour plot, and it's 7 days of data for each patient that's then averaged on top of each other, and it's minute-by-minute data. And the blue line are the healthy match controls, and the red line is the patients. And what we observed, in this case, we didn't look at steps, we looked at a measurement known as activity counts. And we saw during daytime, there was a statistically significant difference between a reduction in activity for those patients. So there was a lower level activity for those individuals with GLUT1 deficiency syndrome. So a really helpful example of how a pilot and upfront study can help you plan for future studies, can help provide the researcher with insight into how to use these actigraphy technologies in future studies, and the sponsor in this case has done in multiple other studies as well. So next slide, please. Well, so one last slide before we turn it over to Abigail, but some I guess, best practices when thinking about designing an actigraphy study or using an actigraphy device in a clinical research study. I think at the highest level, the most important item is make sure you're matching a device to the study. Depending on the goal of the study, the patient population, the visit schedule, there will be different devices that will be better suited for different studies. You really want to think through things such as battery life, memory capacity, ease of use for both the patient, and that will allow you to really deploy a device that will enable strong data collection in each study. And then finally, it's really important to go in when using an actigraphy device with an open mind. I think actigraphy devices is certainly rapidly increasing in a variety of studies, we're still at the early days for use of these technologies. So there's a lot of opportunity to include them, and there's also a lot of benefit from the data that can be collected with these technologies. I would also say that regulatory agencies and the FDA in particular are really excited about what can happen with these technologies and how they can be used, especially in times of COVID. Shifting data collection to the home, something that people are very excited and interested in. And then finally, you really want to, again, be partnering with individuals that are experienced here and can help you plan well so that you can successfully deploy these technologies and collect this data successfully. So I think with that, I'll turn it over to Abigail.
Hi, I'm just going to go over some best practices from the site's perspective to help ensure that the study runs smoothly. My first point is to develop patient engagement strategies for physical activity and activity data. The first thing would be to remote data collect. I call my patients before they start their monitoring period just to see what their schedule is like. I know they gave a list of all the different types of actigraphies and the devices and how they transmit the data. Some of them might need to be a manual upload, and you might need to talk to your patients about when can we decide a time that every day you guys send the data over to me. And then carefully design patient reminders. So in my calendar, I set a reminder to tell my patients to start their monitoring periods. And we have a conversation about the dates that they will do it. And then once you put that into the system, every single day that they're in their monitoring period, you will get email notification essentially telling you how your patient's compliance is doing. And the compliance emails are super helpful because it tells you essentially who you should be inspecting more often. Offer hypercare to clinical research associates, CRAs, and monitors. So the monitors are allowed to do the trainings as well to familiarize themselves with the Actigraphy device. And essentially they can act as a training for you if you have any issues. Also, for me, they can act as an enforcers. If I ever am neglecting a patient and not looking at their compliance, I will get a phone call from my monitor saying, hey, X so-and-so isn't clearing their device, and I will have to contact the patient then. Next slide. Show the data to the coordinators based off of what they need to see. I'm not overwhelmed with all the data points. I see what I need to see. And essentially this helps to keep me blinded. Say for instance, this is a drug you're experimenting with. I won't see the endpoints and assume a patient is on a placebo versus the drug itself. Preparing study sites for using and downloading new technology. The big recommendation would be to download the app itself and familiarize yourself with it. Before I started, I actually called them in advance, and when I knew my patient was on the way, through the phone, we set up the device, and I even walked up and down my aisles and see my steps were calculated. So that was really helpful, but now once you get the hang of it, obviously I don't have to keep doing that. Offer robust support and help desk tools to investigators and site staff. So as soon as you assign a patient to a device, you get an automatic email of a real person essentially asking if you need any assistance, or they will tell you directly if you did a step wrong. They also have a 24/7 help desk where you can call at any time, and they will help walk you through any issues. The advantage of this is it helps protect the patient's rights because they don't contact the patient directly. They will go through me, and then I will contact the patient to troubleshoot some issues. Next one. Kind of already touched on the compliance reminders. Pretty much every day you will get a reminder of how the patients who are in their active monitoring period, how they are doing with compliance. And then troubleshooting for difficult patients. At my site, we have a pediatric study with kids with mental disabilities. We have ran into a lot of issues with this as far as compliance because the children aren't keeping the Actigraphs on their wrists. So we quickly found out that the same data could be collected on the ankle and around the belt, which really helps a lot. And then also it doesn't have to be directly touching the skin. We had one parent who actually wore PJ pants and put the Actigraph in the back pocket and still collected the data for us over the night. And also it's waterproof, which is good for children. That's it.
Chris, did you want to start the QA session?
Yeah, thank you. Sorry, I was just coming off mute. So thank you very much to all three of our presenters. Thank you, Abigail, for the frontline perspective especially. We have time for questions and answers. You're welcome to post those in chat or raise your hand, and we're happy to take them. I have an initial question for Rob, or I think I will point this at Rob. Rob, could you speak just briefly about some of the potential for these devices to measure things that go beyond sort of movement step count activity level? What about the potential for these technologies to interrogate the symptoms of a disease that might be things like a gait disturbance or a challenge in balance. Can you talk to that a little bit?
Sure. So we've done a lot of work on building out validation of empirical measures, walk, speed, distance, and so on. That's what I call the foundation. Where you take it beyond that is to do what Chris speaks to. If you have analytical validity for these measures which can be verified, you can start to look at things like symmetry between one side of the body and the other, variation in lateral stability. You can also, in other ways, use a wrist-worn device to detect rotation of the wrist. An accelerometer is an accelerometer. The measures that you derive must be patient relevant, but it's when you have that kind of data and you signal process it appropriately, you can gather lots of additional information. Would you like me to add any more, Chris?
No, that's very helpful, Rob. Another question that's coming up, Matt, I think maybe this is probably better for you to address. Can you speak a little bit about considerations around patient satisfaction with using these devices? I imagine there's a number of considerations, one of them even being to what extent is the use of one of these devices perhaps kind of expose a patient to making it obvious to people that they're participating in a clinical trial because they're, it may be something that is sort of out of place in their normal routine. And I'm sure there are other issues around that you and your team address.
Yeah. So when it comes to patient satisfaction, I guess I'll unpack that a little bit there. Ease of use is something that you really want to focus on and making it kind of streamlined for the patient. So, a device ideally will have a long battery life, will have good water resistant properties, and allow the patient to kind of to wear it without having to worry about charging it too much and things like that. And when it comes to like signaling to the world that patient is participating in a study, I would think the devices that are being used more commonly have a much more streamlined look and feel than the devices of a previous generation. But in addition to that, I think what's interesting is how some of the I guess what would be known as the consumer grade devices are becoming more clinical in nature and focusing on a lot of different research, whether it's the Apple Watch or a Fitbit or Garmin devices. These devices, fit into everyday life very, very easily, and they are becoming more, more valuable when it comes to research as well.
Thanks, Matt. There's another question that's just come in. It could be for you or Abigail, or you could tag team it, but what are some of the challenges you face with collecting data in the community, and how do you overcome them? I'm interpreting that to mean, so obviously the studies that most of us are referring to here are sponsored studies, and so they have active monitoring. But at the same time, we are putting these on patients in some cases for very long periods of time. So what are some of the mechanisms to help encourage participation? Maybe Matt, if you want to continue since you were just speaking, and Abigail, if you have any additional thoughts.
Yeah, I think, once you send a patient home with a device, the goal is that they'll wear it. So what are methods or tools to ensure that they will wear it? One, you want to explain to them that the importance of wearing this device and kind of the goal of the research. I think in different patient populations, you get really engaged populations who are excited about wearing this device and contributing to research. So there's kind of an educational aspect of it that comes up front. And that education is on both the sponsor and the vendor to support the site, so they feel like they can effectively articulate how to use these devices to the patients. And then I think we touched on some of these kind of tricks or methods of ensuring that we get good data once the patients are wearing the device in the field. And maybe Abigail can touch on this, but compliance alerts via email, really allowing the site to focus on the patients that are having trouble or are not wearing the device. And you want kind of smart alerts that tell you what's going wrong versus just kind of blanket statement every day with an issue. Those are really effective in allowing the site to engage with the patient, engage with the family, and see why the patient wouldn't be wearing the device. I don't know, Abigail, do you have anything else to add there?
I was just going to add something quick that it's essentially wearing a watch, and I'm finding that a lot of my patients don't even really notice it. It's not inconvenience for them. They don't have to really sync it. They just have to be in close proximity of the hub for it to sync automatically. So it doesn't take that much time out of their daily lives to do this.
Yeah, that's really, really key. Sort of easing the friction and trying to make it as natural as possible. Want to see, was there another question that popped up, or did I answer it? We did. Okay, well, thank you all very much to our presenters. Thank you to our participants. Oh wait, we have one more question. Hold on, and this one we absolutely have to take. Jill Mayhew from Ultragenics, who authored a fantastic poster that we referenced with GLUT1 deficiency syndrome, somebody I'm really thrilled to have worked with. So she's posing a question for us. Jill's asking, are there new ways to incorporate event markers that don't necessarily require pushing a button on the sensor? Rob, I think that'd be a great question for you.
Yeah, sure. So we work in this capacity now with many clients. Let's use an example like migraine. In migraine, there's a prodrome that happens before the headache, and a number of migraine therapies are tied to detecting that prodrome. So what we do more and more now is layer on additional event markers. It could be a diary, it could be methods of classifying the data, so that we can start to pick out these key events in the continuous data and then isolate them versus the background data that's important but not relevant to the event. I think that's going to become an ever more important area of research and development. It's something that we work on quite a lot, because you can't, if you want to start to get to fine-grained measures or event-based measures, being able to do this is a must.
It's a great question, and thank you for your response there, Rob. Matt, we have some more questions coming in, so I think sometimes they just take a little time. And maybe, so this question, can you speak a little bit regarding the strategies you implement for patient engagement during trials? Matt, I'm wondering if you can incorporate some of your experience in how we've even seen incentives used to help drive this, particularly across different types of populations and durations of studies.
Yeah, sure. I think there are a variety of methods of keeping patients engaged in a study. So I think in the easiest way, oftentimes if a mobile device of some sort is included, having friendly reminders, whether it's during those specified wear periods to wear the device, is something that works really really well. Other things of encouraging patients to participate in wearing the device, and we've actually gone as far as rewarding patients financially with different levels of compliance. So if patients are able to and are willing to continue to wear a device for X amount of time, obviously you go through the appropriate ethics committee and IRB submissions, but you can give patients the opportunity to be financially rewarded. And one mechanism that we haven't necessarily deployed to date, but something that we consider often, and it's a balance of exposing a patient to their own information but you also want to be blinding them to the treatment. So there's a lot of discussion with sponsors, and I think this is increasing, is being focused on patient centricity and wanting to be transparent with the patient. Perhaps at the end of a study, you report out maybe a holistic view of their activity and sleep data in some sort of PDF report is something that could be used effectively to engage patients to continue to provide data.
It's a great example, Matt, where I think a key that you noted, providing it at the end of the study, you have the opportunity to preserve the blinding and make sure that you're not influencing patient behavior, but at the same time providing that feedback. Patients generated the data, they ought to be able to see it wherever we can reasonably make that possible. Okay, thank you all very much. Again, I think I will really wrap up this time. Appreciated the engagement of the participants, some great questions. And we will make this available on an archive. So if you have colleagues who benefit from it, we'll encourage you to share it. Thank you very much and have a great rest of your day.